Hello, everybody. Greetings from Taiwan and welcome to Prague. Sorry that I was not able to come personally, but at least I can connect online and show you some results of the research I'm doing here, which is focused on forest leaf litter beetles, including staphylonoids. It's originally the idea which I got together with Fangsho, but there were more people helping to us, especially JP, who provided his lab, Han, who provided her knowledge about the next-gen sequencing, and Howard, who helped me to document the specimens. So all that basically started with what we all love, that means sifting forest leaf litter, because we know that the fauna is incredibly rich and there are all these staphylonoid groups living there, right? But if we ask how rich it really is, to answer this question is actually very difficult. There are some first kind of data accumulating recently, but in reality, we don't know. And that's because it's really hard to identify all the species in the sample, even all the beetles. And we usually ignore the larvae, which are naturally even more impossible to identify. So in the beginning, we actually discussed with Fangshuo and we realized that we cannot even answer some basic questions. For example, if we have a one leaf litter sample and we extract all the beetles, like what you can see on the screen, how many species are that? Are there 20, 30, 50, whatever? And if we actually ignore all the larvae on the picture in the sample, do we bias the data which we will get? And maybe more importantly, if we take this such a big leaf litter sample and we get all the beetles, do we document the local species composition, local fauna somehow properly? So to test that, we selected the Huaishun Forest Reserve, which is the central Taiwan area, not really high altitude, but already quite nice, steep, nicely forested mountains. And in this small reserve, we selected three localities lying relatively close to each other, which we dubbed highest, middle, and lowest. And they actually differ in the type of forest. So we try to select as different places as possible to kind of sample across different types of habitats. So the highest locality is actually kind of old secondary forest, very nice, very far from tourist traffic. The middle locality is regrowing cryptomeria plantation, so it's now conifer broadleaf mixed forest with some kind of hiking tourists around. And the lowest locality is basically very horribly looking, disturbed secondary forest with really tons of tourists because it's laying just next to the access road to the area. And so it has also some things like invasive ants, for example. We visited the place 6 times. So each of these three places got 6 samples. So we had 18 full samples in total. We naturally sifted in the normal way as we do. We selected some nice accumulations of leaf litter. We only standardized for volume to have somehow more or less same amount of leaf litter in each sample. So from each visit, we brought three big bags of leaf litter. We processed it with Winkler funnels, as we all do. For 3 days, we changed it twice per day, sorted all the beetles, including larvae, and then sorted all the specimens to morphospecies. Then we moved to Taipei to JP's lab, where we sequenced 1 to 2 specimens per each morphospecies and sample. We used the DNA barcoding gene, and in total we extracted something like 1,134 specimens, of which 965 provided good sequences which were really belonging to the beetles. So once we loaded it into the computer, we got a huge tree which I cannot even show you in whole. So this is not even a half of it, but I can show you a piece of that. And this small piece, you can see that some specimens are green, some are blue. The green ones are larvae, the blue ones are adults. So one thing which is nicely seen here, even that DNA helps us to associate adults and larvae. Also, it helps us to somehow guess how many species we have in the sample. So here you can see that the Anopheles species grouped in 5 kind of nicely defined clades. So we can guess that there's 5 species candidates. We call it OTUs operational taxonomic units. Candidates, because this is only based on the DNA data and they can still be something problematic inside. You can, for example, see that the species B has still some structure inside. So either it can be like a complex of multiple species, or it can be one species with some structured populations. Who knows? We need specialists for that. So Jared, in this case, and check the morphology and trying to figure out. So here you can see some of the specimens, so representatives of all these five Anatolus species, including the larvae, which we associated with it. You can also see that some of the specimens are a little bit broken. This is especially for the specimens we collected in 2019, but nowadays we are extracting in much more careful way. And also, for most specimens, we actually have additional specimens which were not touched. So we have still lots of very nicely preserved specimens to be studied. Sometimes, naturally, it does not go that nice. Once I ran the tree for the first time, I realized that there are two species of Tolmerinus, for example. But Fang Shuo protested. He said, like, no, no, I checked these specimens and they are really the same. Even the genitalia are the same. So I double checked the sequences and I realized that, oh, actually, three of them are not the beetle sequences, but the sequence belong to the terrestrial isopod, a common species in Taiwan. So it looks like that these three beetles actually ate the isopods, and we by mistake sequenced the food instead of the beetle. But at least it can say something about the biology of the beetles, so probably the tolmarinus are kind of partly at least eating the crustaceans. So what is your guess? How many species we had in one big bag of leaf litter? 6 liters of leaf litter. So on average, once we kind of count all these 18 samples we took, we have 58 species of these 23 staphylinoids. Once we consider both adults and larvae, and 45 species, of which is 21 species of staphylinoids when we only count the adults. But more importantly, so how many leaf litter beetles are actually living in the area, right? So for that, we actually have some ways. One of that is so-called species accumulation curve. So we basically can add more and more samples or more and more specimens and check how many more species we are observing once we are adding samples of specimens. From this curve, we can then kind of predict how the curve would continue, and this would kind of tell us what is the expected total species richness in the area. Another approach is to check the rare species. The logic basically is that the more rare species we have, the those only collected in one or two specimens, the more unseen species are there still in the area. So once we did it for all the beetles, we actually sampled 319 species for all the three places, all the 3 years of sampling. But these curves actually show us that the estimate of the local richness in this area is something like 500 to 600 species, which was actually quite shocking because when you compare it how many species we have in one sample, you can see that it's kind of very small proportion. Ignoring larvae, which is the red curve in both graphs, is moreover resulting in bias in kind of underestimating the species richness, which is quite relevant. However, if we check staphylinoids, so naturally we have less of them. We have 160 species in total sampled. Uh, then these curves estimate about 220 species living in the area. Okay, so we are still not totally complete after visiting or after taking 18 samples, but we are doing a little bit better job than in complete beetles. And here you can see that these both curves, so those once we include both larvae and adults and those based only on adults are basically overlapping. So that basically means that that ignoring larvae in case of staphylinoids does not really change our idea about species richness. That's probably because the larvae and adults live both in the same environment in most of these species. So once we are ignoring larvae, sooner or later we are actually collecting the adult of the same species and we build up the kind of result anyway. So what does it mean actually? The main thing for me, the most shocking thing for me is so when you take a 6-liter sifting sample in a forest, then you probably sample around 10% of local leaf litter beetle richness living or leaf litter beetle species living in the area. It's very small amount, actually, small proportion of the local diversity. So what to do with that? So we can actually sample multiple times, right? So this will a little bit help. Or we can actually sample or take multiple samples in one area in one time. We actually did it kind of accidentally because during the first visit in 2019, we actually took 5 samples and we sampled 5 different sites, not 3. This was because we are kind of exploring the area and trying to figure out which 3 places we will sample for longer time. We sequenced all these 5 sites. So we actually, for this case, have the data for 5 sites. We are not estimating it. We just have really the raw data. And from that, which are the curves here, you can see that once we sample one bag of leaf litter, one place, we sample around 10% of the local diversity. But if we sample 5, we sample around 35 to 40 percent. Okay, it's not even half, but still it's better. And in that one day in 2019, we actually collected all these 5 samples in one day. So it's still better to do actually more places in one area than to just take one big sample, which naturally is what we mostly do. Here you can see the number of species per family. So naturally, Staphylinids were the most diverse as it concerns species richness. You can also see how many species we collected only in adults. This is the blue part of the bars. How many we only collected in larvae, which is the orange part of the bars, and how many we collected both in adults and larvae, which is the gray thing. You can see from that that in most cases we collected adults. There are just few families like Tenebrionids or Elaterids, Lycids, Lampyrids, in which most species naturally are in larvae. Another shocking thing, the second shocking thing for me was how few species we actually collect in adults and larvae together. So for all the 319 species we sampled, only in 43 of them we collected both stages, not necessarily in one sample. For staphylinoids, we only collected both adults and larvae from 25 species of staphylinids and 2 species of Acatidium. So really a very small proportion. Here I can show you some lucky ones, so some lucky findings where we actually collected both adults and larvae. So these are the photos of the molecular vouchers. That's why they are not really super perfect, but the specimens are available for study, and we have additional ones. So there are some allocarines, including the phylagrians, which probably don't have the larva described. For pselaphines, actually, we did not collect any single larva. That's really weird. But for sidmanides, we at least get the larva of this weird euconus. This like ball-shaped thing in the abdomen is very characteristic. Pavel Yaroshinsky already has this larva and is working on it, I believe. For osorines, we succeeded to get larvae for 2 species. In this Thoracochirus, the weird thing for me is look at how the intestines goes inside of the body of the larva. It goes something like a back and forward and back. It's kind of coiled inside, all over the body. I don't know if this is something normal for staphylinids, but for me it looked very unusual. For pederines, we have also some species, including the Homeotarsus, which probably also is the group which doesn't have the larva officially described. Then we have some Scaphizoma, 2 species, the Anotylus, which I have already shown you, 2 species of Tachyporines, and some species of Staphylinines, including the Diocus, Tormarinus, some Quidius, some Ericksonius, and so on. And actually many more, which we have no time for now. But we are already kind of having all the photos in a folder, which I can easily share with you. For staphylinids, we already photographed all species, and for the other groups, we are basically just adding it right now. all the material is now sorted by these numbers, right? So we have all the species candidates are numbered. We have them all together, and then we naturally have all the additional specimens safely in the fridge in the very good condition in alcohol still. And we would be naturally very interested and very happy for any help with identification or any cooperation in kind of more detailed way on systematics of these things, larval morphology, biology, and so on. We can borrow the specimens for the study. We can share part of them. Holotypes that need to be deposited in Taiwan, but otherwise the material is quite very free. And the next steps, so what's to do in the future? So we want to continue the similar way across Taiwan. So this, what we did so far was basically just one place. And now we are interested what will happen once we will start to sample different places across the complete island. Then my dream is to build up the online freely accessible DNA barcoding database where we can actually put all these DNA sequences and all this voucher information. And I definitely want to publish kind of report on that as a normal publication using or including all the people who will help with identifications as co-authors. And then naturally, if there are specialists for the particular groups who are interested or who can help us at least a little bit with it, I would be really happy to start detailed treatments on systematics and larval morphology and biology of these things. For example, we already did that for the Oxyomos, which is the aphrodine living in leaf litter, which we collected in the same place. This was because of Bing Hong, who is the local scarab specialist and who was happy to help us. But I hope that actually we can find more people, including some of you, to help us with some kind of these more detailed treatments, and we can do more for understanding Taiwanese leaf litter beetle diversity. Thank you very much for your attention. So this was supported naturally by many institutions, by the Taiwanese Ministry of Science and Technology. I really hope that we will also get your support. Thank you very much.